Merci, Gaël. I want to start by thanking the organizers for giving me this opportunity. It's my first time in China. It's not my last time, I can tell you this. Yeah, so I'm going to do something a little different today. It's a keynote, a little bit more time, so I'm going to be probably over-ambitious, but I'm going to try to give you a reflection, historical reflection on how the perception of transposable elements has changed since their discovery by McClintock. So I'm going to try to cover a lot of ground. And I'm going to emphasize some key words that you all know, but perhaps you don't know the historical context by which these words were first used. But I think it's very interesting to reflect on how the choice of certain words that we use as scientists has such a large impact, when in fact, at the end, they're just words. But they're convenient. And you see, it's going to become clear what I mean by this as we go through this talk. Okay. So let's start with the beginning. Beginning, obviously, is Barbara McClintock, my hero. So as you probably, most of you probably know, Barbara didn't call transposable elements, she called them controlling elements, because she thought that these elements were actually the mechanism by which genes in eukaryotes, in maize at least, she was studying maize, of course, were regulated, were being turned on and off during development. This is really one of the key questions she was after. Of course, also, in doing so, she realized that these elements were mobile genetic elements, in addition to their ability to modulate the expression of genes. But really, I think what was really driving her work at that time was this idea that these elements were switches, were normal components. She called them normal components of the chromosome. Remind you, this is 1956 or in the 50s or late 40s, so this is prior to the discovery of the lac operon by Jacob and Moe, so we know nothing about how genes are being turned on and off in any organisms. So she thought that they were responsible for controlling differentially the time and activity of individual genes. However, she didn't receive the Nobel Prize for that discovery. She received the Nobel Prize for the discovery of mobile genetic element, which sure enough was a very spectacular and a breakthrough in genetics. Now, in the early 80s, in 1980, in fact, very important pairs of conceptual papers, theoretical papers, very short, 2 papers published in Nature came out in 1980 that bring the concept of selfish and parasitic DNA. These 2 papers by Orgel and Crick and Doolittle and Sapienza were published back to back. I really encourage you to go back and read them. They are really fun to read, and they're really interesting and insightful. And what they proposed is a very radically different version of, a very different view of looking at transposable elements than McClintock was thinking about. They were thinking about these elements as parasitic elements and selfish genetic elements. And first, they borrowed this concept from Richard Dawkins that came up with it before, although in a very kind of different flavor. And they stated in Orgel and Crick stated in this paper that when a given piece of DNA or class of DNA of unproven phenotypic function can be shown to have encoded a strategy such as transposition, which ensures its propagation, its genomic survival, then no other explanation is required to explain its existence. Okay? And they added this sentence, which I think, in retrospect, I think the reviewers should have asked them to take this out because it didn't bring much and was fairly derogatory. But it says, the search for other explanations may prove, if not intellectually sterile, ultimately futile. So basically, we're all but saying, if you read between the lines, that this is not that interesting maybe to study, or at least not the way you're thinking it is, not as normal components of the chromosome. Okay. So these papers had a major impact on how we think, and still today we think, and they are a very useful concept, and they are backed up by a lot of data that supports this idea that transposable elements are parasitic. In particular, studies in the 80s and 90s were dominated by the study of active transposable elements and their mutations, the mutations that they create that were visible in all the model organisms and many other organisms. And in fact, it's really cool because you can rewrite the history of genetics or part of the history of genetics by looking at transposon-induced mutations. There's only a few here to illustrate this point, but Mendel's rankle P was a transposon insertion indeed. The white eye mutants, many white eye mutants of flies were transposon insertion. Hox genes were discovered because of transposon rearrangements and insertions as well, and the list goes on and on. Now, those are all mutants, and as you can appreciate from these pictures, these phenotypes are very likely to be fit for to favor the fitness of the organism. So they really was consistent with this idea that these elements are potent mutagen and deleterious mutagens. And Kathy illustrated this point very, very nicely in her talk. Always a very hard act to follow, by the way, but I'm going to try. Of course, this was further later demonstrated in humans, where Hekazazian and colleagues isolated the first active de novo transposition events in the factor VIII gene in 2 boys, if I remember correctly, that would cause hemophilia in these boys. And today we know of over 100 human genetic diseases that are caused by de novo transposable insertions or chromosomal rearrangements that they promote. So clearly all of these examples that I'm showing you here illustrates this very potent mutagenic impact of transposable elements. And again, Kathy really gave you a full tour of all the things all the gymnastics that they can promote and how the different ways they can break chromosomes and affect gene expression in many different ways. Now, then this illustrates obviously the idea that these elements, if not controlled, are deleterious. Often their insertions are. Of course, we also know, in fact, this is backed up by population genetics in humans and flies, that probably most insertions are neutral, especially in humans, actually, when there is a lot of room to insert in a sort of quiet fashion. And of course, on occasion, although probably very rarely so, these new insertions in the genome are unlikely to be beneficial to the organism, although we know now a few examples of this. For example, the pepper moth and the poster child of the adaptive evolution during the industrial revolution, the melanism of this moth in England was driven by a new insertion that was obviously adaptive. So there are a few examples out there, but very few. Okay, so this deleterious potential, I would argue, then it really implies that you're going to have coevolution between the host and parasite. Otherwise, if you don't do that, you will eventually kill the host and kill yourself. So transposable elements have evolved actually a battery of mechanisms to self-control and minimize their deleterious impact on the genome. I'm not going to cite too many, but for example, many elements have exquisite target specificity, for example, in yeast, where they go and insert in a spot in the genome that clearly is a safe haven in the genome, doesn't affect gene expression or anything. And they do so by having evolved very sophisticated co-evolutionary interactions with host proteins that mediate this targeting. That's just one example. Also, many transposable elements are suboptimal for transposition. You can make them better in a test tube, or through mutagenesis experiments, especially DNA transposons. Every single time people have tried to make them jump more, you can by making mutations in them. So they're sort of suboptimal. Okay. Now, on the host side, there's also many control mechanisms that have evolved, and we're going to hear many of them at these meetings. RNA, zinc fingers, proteins. Some of these mechanisms look like they are dedicated systems to control and suppress the activity of transposable elements. This is also consistent with the parasitic DNA IDE, where you will evolve mechanisms to minimize the deleterious impact of these elements. But then I would argue that this coevolution, in fact, has really enabled transposable elements to propagate and diversify enormously in genomes. It's because precisely of all these control mechanisms that they can proliferate, and in fact it drives their diversification themselves. Now, we also understand now that most new insertions would be eliminated by natural selection, even if they are neutral, just by genetic drift. And also under genetic drift, a subset of these elements will reach fixation, even regardless of whether they have a deleterious impact, or even neutral. Once fixed, then, the idea is that most T copies would then be repressed, those that survive selection, if you want, and would not contribute generally to host thickness, and they would progressively decay by point mutations. And this idea leads us to another key concept in the field of junk DNA. That's what the concept of junk DNA is about. This is a very useful concept as well. And so it takes us now to genomics, because this is when this concept of junk DNA become really obvious, when people started to sequence whole genomes, starting with the human genome or the fly genome. It was very clear that genomes were filled with dead, decaying transposable elements. This is where I entered the field. Okay? I started studying transposable elements in the beginning of this genomics era in the early 2000s. And I was a postdoc in Sue Wessler's lab, and I wanted to put a slide that I found in my archive from Sue. This is not my slide, that she put together when the maize genome was started to be sequenced. So the genome where McClintock found transposable elements, it was very clear that the elements that McClintock was studying, like AC and DS, were very rare. They're very low copy number, so the active elements, the ones that actually jump. Instead, the maize genomes was a graveyard of transposable elements. She called it an ocean of transposons on which tiny gene islands are floating. So that was the picture of the genomes that we're starting to get from this sequencing. It wasn't a complete surprise, but when you start seeing it and unpacking all these layers of dead transposable elements, it was becoming really obvious that this is what most genomes are made of. Now, this is a picture of me back then, way back, at the time where I was just about to start my own lab, and I use this as a transition, because we moved to study the human genome in my lab. And we wanted, in fact, we wanted to find that there were more active transposons than people thought. And in fact, we were looking at the DNA transposons that Kathy talked about. And actually, we came back empty-handed. We didn't find them. It was indeed, the human genome was largely a graveyard of dead transposable elements. And this is a picture from the Lander initial sequencing of the human genome. This is the paper that you absolutely must read when you start coming to my lab. In particular, of course, the transposable element sections that John contributed to, and Ariane Smith made major work, major contributions, very, very, very influential study for me personally. And this is the famous graph that depicts the demography of human transposable elements. And we're not going to get into the details, but this is a breakdown of the composition of, as you know, half of the human genome, as Kathy explained, is made of transposable elements. We're looking at this half of the genome. And we order them by how old these elements are, basically by how much they've drifted, they decayed from their ancestral sequence, their consensus sequence. And you can see here, most of these elements are very degenerate. In fact, they're not that close to one another. People think, oh, they're not mappable, blah, blah. No, most of them are actually very much mappable because they're highly decayed and highly diverged from one another. They're very old. And really, 99.999% of transposable elements in the human genome are dead, transpositionally dead. Only a very few are still capable of mobilizing, and these actually will be in that very last tile here, okay? Okay, so this is really, I think this concept of junk DNA is extremely clear in the human genome because all these mutations here largely tick at the neutral clock. This has been measured, this has been looked at. For the most part, they're really neutrally decaying. And every species out there has a different graveyard of transposons. So that's kind of what makes it fun. And I put this slide because these are some of the genomes that in my group we've been lucky to work on over the years. It's extremely fun to go into a new genome and find what kind of transposons they have. They all have different transposons, but they're largely dead. Now, the level at which the transposable elements are dead in the genome is extremely variable. I want to emphasize this. The human genome is perhaps an extreme example. If you look at the fly genomes, you will find that there's a lot more active transposable elements in there, okay? So I'm oversimplifying a lot of things here. Okay, so yeah, this is just a picture for fun. Some of the creatures we worked on, and actually I added also this picture because I was very happy to see. This is one of the beautiful things with conferences, you see people you haven't seen for ages. And today I'm very happy that in the audience there's Sun Chang, who was a postdoc in the lab back when we were in Texas. And this is a picture of the lab back around 2010, at the time where we're mining all these different genomes and so on. Okay, so this is the picture that we had of transposable elements, I think, in the early 2000s. Genomes filled with dead Ts. Now, the concept of junk DNA is useful because it's distinct from trash. What's the difference? the trash, you throw it away. You just get rid of it. The junk, you put it in the attic in case maybe one day you may want to go back and reuse it. So that's an interesting analogy. And indeed, this is what us and many others have been looking for. They've been looking for like gold in the middle of the trash, in the middle of the junk, or potential gold. Again, this concept is not new, and ever since the human genome was published, was released, and other genomes, people started to find where are the needles in the haystack? Where are these like nuggets of transposons that have been co-opted now? We're talking about co-option. At this point in time, these elements are no longer transposable. They've transposed, but they're not transposable. Okay, you see the difference. Okay, so you can turn them into gold in many, many different ways. I'm certainly not going to cover this whole entire area. We're going to hear many stories at this meeting, fascinating stories of co-option, but they fall into two major flavors, the regulatory ones and the regulatory sequence and the coding sequence. So the non-coding part of the transposon sequence or the coding part. Now, as far as the non-coding part, what I'm talking about here mostly is sequence that are bound by transcription factors that become regulatory sequence or cis-regulatory elements. You can also see this at the RNA level, but I'm not going to talk about this, but cis-regulatory DNA. So where are we with this concept? Well, it turns out it's not uncommon. I can tell you that much. It's actually pretty common for transposable elements to donate cis-regulatory elements to the host. This is a summary of a study we did with Ting in collaboration with Ting as part of the ENCODE project over the last few years that sort of tried to catalog and quantify really how many cis-regulatory elements in the human genome are derived with confidence from transposable elements. And I'm giving you the punchline. You can look at all the details in the paper. We do a lot of detailed analysis of these elements and where they come from, what kind of transposons, how do they acquire or lose some cis-regulatory activities. There's a lot of intricacies I'm not going to get into. But the punchline is 25%, that's about 200,000 cis-regulatory elements predicted by ENCODE today, are derived from transposable elements. We can say that with some level of confidence. So that's a lot. Even in my wildest dreams, I didn't think like a quarter of our regulatory elements would be derived from transposable elements. Oh no, I'm not saying that all of these elements are functional, okay, don't get me wrong. But at least a quarter of the ones that we think are functional, they are in there, are derived from transposable elements. And of course, there's an increasing number of elements that have been characterized, that have been deleted by CRISPR. We and others have done this. We're going to hear stories at this meeting, I'm sure, that validate the cis-regulatory activity of these elements. So it's not an uncommon phenomenon. And it's not restricted to humans or even mammals. There are beautiful examples of cis-regulatory networks, entire networks of cis-regulatory elements that have been dispersed by transposable elements in all the model organisms today and other species. Now, when we come to the coding sequence now that are derived from transposable elements and give rise to new genes, new protein coding genes, we've also been very interested in this concept over the years, and there are also now a growing list of genes that we know are derived from transposable elements and have important functions. This is the idea of genes being born from transposons, so moving from the dark side to the bright side now. Instead of being a mobile gene, a parasitic gene, you become a host gene that's immobilized and now serving a cellular function. Why genes from TEs? Well, there's a number of reasons. First, transposable elements are extremely abundant, extremely diverse. They are mobile, so that's a very interesting feature to create a new gene because you introduce a new sequence, a new coding sequence, protein coding sequence in a new genomic context every time you have a new insertion. And because these elements often carry splice sites within them, it's a very convenient way to create new gene architecture by alternative splicing and picking up exons from transposable elements. And we and others have described now hundreds of cases by which genes were born this way by fusion with other existing exons. I'm not going to go into, you can't see this because that's on purpose. It's to illustrate that there's a great diversity of protein domains. And in fact, now we know that every known activity of a transposable element has been at least once co-opted for cellular function. Reverse transcriptase, that's exceedingly rare, but there's a few examples out there. Much more commonly, transposase and DNA-binding proteins are created by borrowing domains from transposase, and also RNA binding proteins often are borrowed their RNA binding domains from GAG proteins. So indeed, in the human genome today, we have about 200 genes. That's a conservative estimate that I have tallied over the years. So about 1% of our genes, I'm talking about protein coding genes, are transposon derived in an unambiguous fashion, if you want. Again, many other examples exist, and I think, in fact, this is still understudied. I think there is probably many more transposon genes out there. Often it is difficult to distinguish a transposon gene from a host gene. It's much easier in the human genome because the elements decay and you can really contrast. There's very few active elements, so they really stand out, these domesticated genes. Okay, so where are we now? So I'm going to talk now about what I think is maybe one part of the future in the field, or at least what I'm excited about these days. But suffice to say, I think by now, I think it's now recognized that transposable elements, parts of transposable elements, are often recycled for cellular functions. And I really want to emphasize this. It's not just the change in the way people think. People start to change in the way they're thinking when they can see things, when the data is, we're scientists. When the data is in front of you, you just can no longer deny it. So it's not just a dogmatic change, if you want. It's really what all of these advances have been driven by technological advances. And sure enough, more and more we can actually see transposable elements. We can study them. We have the tools now to do it with unprecedented precision. Certainly, long-read sequencing is another really transformative way by which transposable elements are being studied. Okay, good. Oh yeah, okay, thank you. So what's next? So what's next for us, at least, is really this question of how do you go from being a parasitic selfish element to a co-opted transposable element? I told you it's pretty common, but how do you transition there? How do you get there? From being purely selfish to co-opted. And I have to say, back a few years back, I was really thinking about this question in a binary fashion. And as you know, in life, nothing is binary. It's always a gradient, it's always a shade. And in fact, this is exactly the idea that Sylvia Cheng, former graduate student, now a postdoc with Feng Zhang at the Broad Institute, brought to my lab. She was like, I think you're not thinking about this right. It's not binary like this. It could be that there's a transitional phase. It could be that there's a phase where the transposon is both wild transposon and both selfish element and still contributing to the host fitness. So I just don't really believe that. I was very, very, very skeptical about this idea of cooperation between an active transposable element and the host. And it took her about 6 years to really basically prove me wrong. And I'm very grateful to her. That's really a wonderful thing when you get a trainee into your lab that challenges the way you're thinking, challenges the dogma in the field, and prove to be right. She proved to be right. So I'm going to give you really the summary of this study because now it's about to come out. There's a preprint out there. You can look at all the details. But I just give you the quick summary. Yes, so now I believe it. Okay, so to prove this, she wanted to find active transposons, and she wanted to find a system by which you could manipulate the elements to see if they were important for development. So Sylvia had in mind that transposable elements, kind of like McClintock was thinking, were essential for development even when they're active and mobile in the genome. And she picked zebrafish. We don't do zebrafish in my lab. So she had a lot of work to do to learn how to manipulate zebrafish. She went to a course, blah, 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 and she actually really honed her skills and to bring zebrafish into the lab, which we study still today. And she started actually by mining single-cell RNA-seq data that was available for zebrafish. Also, she wasn't trained as a computational biologist, by the way, so she also learned all of this on her own. And she found several very interesting candidates. So what we were looking for? So we were looking for elements that actually would be expressed. So first, we were looking for active elements, or elements with a hallmark of active transposable elements, okay, not dead elements. And then she was looking for elements that would be active in somatic cells, somatic lineages during development, because it's not really exactly what you expect for a selfish element to be expressed. you expect it to be expressed in the germline. So we thought if we see also an element that may be expressed in germline, but also in a somatic lineage, that might indicate that it has taken on some sort of function in that lineage. And she settled on this one element, bicary. We didn't name the element, he actually been discovered before, because it was really a striking marker of the mesendoderm, early mesoderm, if you want, of the zebrafish embryo. So this is a single-cell RNA trajectory kind of analysis. And in fact, it's a sophisticated way of rediscovering something that had been discovered, in fact, 20 years before. This was an element that been pulled from a screen looking for markers of mesendoderm in the zebrafish embryo. However, they didn't know what kind of transposable element it was. They knew, they called it bicary. They knew it was a transposable element because they knew it was there was multiple copies in the genome, and they were flanked by long-term RNA repeats, LTRs. But the protein coding sequence had no homology at the time with no other sequence. So they didn't really know, but they used it as a marker in developmental biology in zebrafish. So that's the element that she picked, and then then we went to look at very closely at this element to first look if it was really very recently potentially still active. And I'm going to just summarize that we don't have actually direct evidence of de novo transposition of these elements. We haven't actually looked for this. We should. What we know is that these elements are very recent because some are identical to one another. They are over 5 kb long, 8 kb long, something like that. They are nearly identical, and they are polymorphic among zebrafish strains, so the insertion is polymorphic. So some strains have variable copy numbers of these elements, which is one of the things that makes this study, I think, even more provocative because these elements are not fixed in the genome. So we have reasons to believe that these are recently, at least very recently active and perhaps still active. And they encode, interestingly, all the elements in the reference genome only encode one protein, so they're non-autonomous elements. They're not capable of making all the machinery. There's probably one out there that is, although we haven't found it yet. They only encode GAG. Okay? So then Sylvia went on and went to manipulate these elements, and I'm not going to show you all the details. Again, I'm going to summarize what she found. She used a different approach, actually, to manipulate these elements in the zebrafish embryo. I need to say these experiments are always very tricky. These are multi-copy number elements. There are actually hundreds of copies of these elements in the zebrafish genome. So to go and affect all of them at once is a messy experiment and a difficult one. She ended up settling the thing that worked the best for us were locked nucleic acid antisense oligos that would promote the degradation of the RNA of these elements. So she injected a lot of embryos, thousands of embryos, and she achieved about 75% knockdown on average across experiments. And then she looked for phenotypes, right? What happened? And basically, most of the time, what happens is that the embryos die. They really don't like to lose for this RNA to be depleted from embryos. Within 24 hours, you got 50% of the embryos that normally survive that would actually die. But if you look at those that survived, and you can look for phenotypes, early phenotypes, and what you see is a typical phenotype in the zebrafish mesendoderm field, which is called the loss of a convergent extension, which is a cell migration process by which the somites of the embryos develop. So you see embryos that are blunted on both ends. Often they have no eyes. They really don't look good. This is very consistent. You can see this. She scored this across many injections. And you can see very nicely the somite phenotypes. If you hybridize for a marker of somite development, MyOD, you can see that the somites in the knockdown embryos are squished like this, basically. They are compressed and elongated in this way. This is actually a quantifiable phenotype. This is going to become important in the next slide. And even if you follow some of the surviving embryos, there's not many, they have this typical tail that curves back. These phenotypes have been described in studies of zebrafish mesoderm development as a failure of early mesoderm development. So I'm telling you that it suggests that these elements are essential for mesoderm development, or their GAG proteins are. What we know is the protein is because she did the rescue experiments. So this is the key experiments that you would want to see in an experiment like this, because you affect many elements. There is many potential for off-target effects. So I only really believed in this, personally, when she brought back the result of the rescue experiments. So in the rescue experiments, she's asked to inject the ASO to induce the depletion of the RNA, and she co-injects a messenger RNA that encodes the full GAG protein, open winning frame. And she has some level of rescue. It's not perfect, not complete, but we were really happy to see partial rescue of that somite phenotypes, which I didn't explain this, but you can quantify it by measuring the length of the somites and the width of the somites. So again, co-injected ASO and the GAG mRNA partially rescues the somite phenotype. Importantly, when she does the exact same experiment, but instead of the rescue mRNA, she uses an RNA that has a single mutation that introduces a premature stop codon in the GAG protein, she cannot rescue. So that is an indication that it's not the RNA itself, but it's the protein that's important for the phenotype, for its function. And finally, she did all of these experiments, not with ASO, but also with morpholinos, and she gets the same phenotypic outcome. That is very important as well, because morpholinos don't work the same way than ASOs. They prevent translation of mRNAs. So that yet another indication is the protein, the GAG protein of these elements that is doing something in development, in mesoderm development. So what does it do? What is the mechanism? Short answer is like, we don't really know. But we have hints from multiple experiments that I'm going to summarize now that it may have something to do with cell adhesion and cell migration. First of all, the phenotype. I told you it's been seen before. It's actually the exact same phenotype. It phenocopies, the depletion of bicary phenocopies the loss of N-cadherin. These are cell adhesion proteins that are really important in the migration of these mesoderm, early mesoderm cells. Also, Van mutants. This is another pathway, non-canonical Wnt signaling pathway that modulates cell migration, have the exact same phenotype. In addition, what Sylvia did, a very cool experiment, she injected the bicary mRNA in a chicken embryo, in a neural crest, specifically of the chicken embryo, and found that it actually modulates cell migration of these neural crest cells in the chicken. So the zebrafish protein in the chicken has this ability to modulate, to oppose actually in this case, the neural crest migration in chicken embryos. We did this in collaboration with Marco Simoes Costa at Harvard, who studied neural crest cells in chicken. When you express bicary in human cells, it aggregates at the membrane. We don't know in the zebrafish embryo because we really failed to get a good antibody for this protein so far. So we really are lacking on in vivo imaging experiments to see really what this protein is doing, where is it localized in vivo in the embryo. But all collectively, these results suggest that the GAG of bicary opposes cell migration and or promotes cell adhesion. Well, you may wonder, is this really a one-time thing, a weird thing about these bicary elements, or is there more examples like this? We think there's more. One was really sitting in front of our eyes was actually another marker in zebrafish. This time, not of the mesoderm, but of the neural crest. It's called crestin. Been published more than 20 years ago as marking very specifically the neural crest. Again, kind of same story, the bicary, they used it as a marker. No one actually manipulated this protein at the time. And they didn't really know what it encoded. But when we looked at the predicted AlphaFold structure of crestin, we realized it's a GAG protein. And when we looked where it was encoded, we found it was encoded by a family of LTR retrotransposon distantly related to the bicary. So it's called bicary2. It's already an annotated element in the zebrafish genome. And sure enough, when we also look at the evolutionary history of these elements, we see they're very recently active, they're polymorphic among strains. In fact, the crestin locus itself is polymorphic in zebrafish. When Sylvia used morpholinos to deplete bicary2 GAG RNA, or actually block the translation of this RNA, I should say this is a morpholino experiment, she could see aberrant cell migration of neural crest cells in zebrafish. So once again, we have a GAG protein that modulates cell migration during normal development of zebrafish. Okay? I'm not going to go through the details. Here you can look at the paper. What about chicken? Well, it turns out people had worked on chicken GAG, but they didn't know they had worked on chicken GAG. Once again, there was a marker of neural crest in chicken that was called ERNI, that was known to be important for the emergence of the neural crest, early emergence of neural crest, neural plate cell. And again, when we looked at this mysterious protein, which in this case had been manipulated in some recent papers and showing that it actually reduces neural crest migration, kind of like the bicary in the fish, we found it was a GAG. It was a typical GAG capsid with a very long N-terminal call domain. Actually very similar in structure to the bicary GAG, but completely unrelated group of endogenous retroviruses in chicken. Okay, they're completely different clade. So now what we have is 3 different active transposable elements, BCARY1, BCARY2, and ERNI in chicken, that encode GAG proteins that are required, apparently essential, for very conserved, crucial developmental process. So this is very surprising, because we wouldn't expect these new elements to do something in this conserved developmental process. So how do we explain these paradoxical results? So we proposed the model of transposon addiction. And the way this is my own contribution, my only contribution actually to this work, is this word. You see, I chose a word. I'm not sure that's a good one, because I'm kind of seeing this as a little bit evil, right? But in fact, you can think about it as a mutualism or cooperation that also works, okay? But what we think is that when the elements first enter the genome and encode these proteins, it really doesn't really matter, because 1 or 2 copies of these elements is probably not going to have much effect. However, as the copy number increases in the genome, if these proteins have a function that is redundant with an endogenous cellular process that modulates development, then you open the door now for becoming dependent on this protein, because what you do here, you might, just like gene duplication, you might relax selection on the endogenous system at a point where now, at some point, you're stuck. You got to start using the transposon-encoded protein to carry out this function. So you may be able to backtrack this, but as the element continue to proliferate, you're kind of stuck. And how do you resolve this addiction? We think one way to resolve this addiction would be now a full-blown co-option of one of these copies in the genome that becomes now under the control of the host, and now no longer under control of the transposon. And that's maybe what we see when we look at these genes such as ARC, these GAG-derived genes that are unique genes, they're single copy gene, that may be sort of the end of the spectrum, the full co-option. So we think that transposable elements can be in that sort of phase where they are progressively, at least, becoming essential for the host. And that's really, I think, a different way of thinking about transposable elements. So I'm going to stop here just to say that we don't think it's an isolated example. In fact, there's people in this room, such as Zizi and Haru Siomi, who are going to tell us maybe stories, other stories of co-option, of addiction, I should say, mutualism of active transposable elements that are also important for the host. So I think the list is growing, and I predict that we are going to see more and more over the years. So I'm going to leave here and tell you that, yes, indeed, now we are returning to McClintock's idea of thinking of transposons are completely integrated within the organism, where you can no longer dissociate them from the organism. So they're not purely selfish, at least sometimes. Okay, so that's the conclusion. You all know how cool transposons are, so I'm going to kind of skip this and all the things they can do. Again, I think that maybe some may be more mutualistic than we think. They may do actually, they may have cooperative activity with the host. But I think overall, I think we will all agree, I'm going to preach to the choir and say this is still, transposable is vastly understudied. I like to say that 99% of people work on 1% of the genome. So I think we need more people in this field. So the most important slide is this one, because first it highlights something that's really actually in danger these days, the funding. All of our work is funded by NIH. Without the NIH, we will not be there. So that they bring us money, and these folks, they bring me joy and pride and inspiration. So with that, I hope we have time for questions. Thank you so much.
Thank you, Cedric. John, you want to ask a question?
Great talk, Cedric. I really enjoyed that. Thank you. I guess I have some questions about your rescue experiment. So I could buy that knocking down GAG the various ways you did it shows different developmental phenotypes. But I'd like to know about the order of addition in that experiment, because I think if you change a developmental profile, I don't think adding the RNA back afterwards is going to be sufficient to revert that phenotype. That's essentially a type of IPS experiment. So how many different ways did you try the complementation experiment? And is what I'm worried about a potential worry?
A potential one?
Worry.
Worry? Honestly, to be honest, I didn't hear half of your question, so maybe not. So
okay, well, okay.
How many times? No, that's the only way we did it. Injecting the RNA. Yeah, that's how.
No, but if you're changing a developmental fate.
The what?
If you're changing a developmental fate by knocking out a GAG gene, I would argue that simply re-expressing that gene is not going to reverse that phenotype. It's essentially an IPS type of experiment. So I'm questioning the interpretation of the results from that experiment. Everything else I love, but I have selfish reasons to want to do this. I would be cautious about the interpretation of that experiment.
Thank you. Well taken.
You want to go?
Hi, Cedric. Very exciting talk. So I have actually two simple questions. So the first one is like, since its background is active transposon, right? Coding a GAG protein. I'm wondering, like, the copy numbers in the genome, since it's active, is transposing, like, maybe in some strains but not others. So, but if it's essential for the development, so do you think the copy number actually matters?
Yes. Yes, thank you for this point. Very, very good point. Yes, so this is with the idea of the addiction, is like it's a dose-sensitive thing. And indeed, actually, the rescue was very tricky because the dose is critical. In fact, if you overexpress this protein, as I've shown you in the chicken experiment, it has an effect. It's likely to be very toxic. So yes, would want to understand the evolution of this. How can you get selection on a multi-copy, element to arrive there? Unfortunately, zebrafish is, we think, not the best system for this because we don't have a lot of genomics resources, so we can't really look at the population genetics of this, which is kind of where you're going, I think. So now we're actually testing this idea in Drosophila, in fruit flies, where there's plenty of candidates actually of GAG encoding elements in development in the mesoderm as well, it turns out. Because we think there we'll get a much better picture of copy number variation and the selection, and how selection can operate at the level of a species in populations to shape this. So it's a really good point. In the zebrafish, it's also really tricky. We haven't been able to test this idea of dose-specific effect. But yeah, thank you.
Okay. So the second question is about the...
If we can just one question, and then we can come back after. Thanks.
Emily?
Thank you.
Oh, no.
Go ahead, Edith.
Hi. I'm just curious, mechanistically, how does the big and all these GAG do to impact cell migration and impact cell adhesion during display determination?
What's the mechanism? Is that what you ask?
Yeah, what is the cellular mechanism? Do they
I wish
package
I would know.
mRNA into intracellular vesicles, or
Right.
I mean, like the ARC story, or do they have something new?
Yeah. I can only speculate again. I told you everything we know about the mechanism. I didn't tell you about all the failed experiments in zebrafish because it's hard to interpret them. GAG, so this is a very recently active element, probably still active element, probably making a capsid. So that part we're pretty sure, although now we're testing formally the fact that if we express this, we can actually see capsids under the microscope. So we don't even know that, but it's a safe bet to think that this thing makes a viral capsid that packages its own RNA. Now, you make capsid at the membrane, typically. So GAG proteins do target membrane through the N-terminal regions, and actually in human cells, we were able to show that, as predicted, the N-terminal domain of the BK GAG is required to see enrichment at the membrane, as predicted for retroviral GAG protein. So we think then making these viral particles, by itself, may actually potentially modulate cell adhesion by itself. That's one idea. That's the simplest idea. So in fact, it's not so much about what the capsid carry or what they traffic or what their cargo is, but it's simply the idea of budding out of the cells that has an effect on cell migration / adhesion, maybe directly affecting cadherins. This has been seen with extracellular vesicles. If you modulate the traffic of vesicles, you can get modulation of cell adhesion / migration. So that's the simplest model, but again, a speculation at this point only. Then we can at the bar, we can talk about more exotic mechanisms. But we think actually this might tell us a little bit, if we continue to study this, which I'm not sure, but it might tell us a little bit about what the domesticated GAG proteins do, such as ARC, PEG10. Those are very interesting. They make capsid in our brain. They modulate memory. What is going on? No one really knows. So we think maybe some of these mechanisms are at play as well for these domesticated GAG proteins. Yeah.
Thank you.
Yes. So I have a related question to the previous one. So you said that there are hundreds of copies of BIC1 in the zebrafish genome. Do you have any idea of which copy, if not all, are expressed?
Yeah, great question. And I oversimplify. There's hundreds, of course, but there's actually only a few dozen GAG encoding copies. In fact, I can't remember the number now. I should know, but I don't remember. Maybe 26. All of the other ones are solo LTRs. As always, with these type of elements, there's an abundance of solo LTRs. And of the GAG coding copies, based on all the data we have, which is RNA-seq data, single-cell RNA-seq data, and also some attack-seq data, which is helpful to pinpoint which copy might be actually transcribed. Sylvia looked into this in some detail. We think that most of these copies are transcribed on their own. So certainly not one or two copies. It's a kind of a global effect, and yes, it goes back to the question of dose. And they're very extremely strong LTRs. They are 1,500 base pair long. They are stuffed with transcription factor binding sites. I'm not going to get into it now. And yeah, so I think there are strong, potent, transcribe units.
Okay, and you don't have any idea about the transcription factor involved in
Yes, we do. I can, yeah. In fact, in the early paper of Bicari, they already identified some path with some factors that modulate the expression of Bicari. So there was a little bit of work already done about this. I forgot to say, my talk is really a conversation starter for the bar. So I'm very happy to answer more questions over there.
Okay. So with this, thank you very much again.